Okie doke. So today, 6/20/2023, the year of our Lord, and we are going to start working on lecture 2 material. So I think yesterday, maybe the day before, I went ahead and opened up the next module because I noticed some of the people were getting done with their test. And just in case anybody felt real froggy and wanted to jump into the next section, I went ahead and opened up this one. But let me go ahead and open up the next two little modules as well. So here and here. So now you'll be able to see where we're heading next. Pretty straightforward with this section. Again, I did modify this from the original syllabus. I did take out one section. We'll do that later. But for now, we're right here, and we're going to be working on cardio phys today. A lot of basic stuff again. Cardio is one of those chapters where it comes up so often that you really have to kind of master the basics before you get any further into it. And once you master the basics, everything else becomes really super simplified. So we're going to do our little circle diagram again. So if you've had me before, this is kind of like our bread and butter for teaching cardio. And it's one of those tools that's easy to visualize and recreate. So if you're taking a test about cardio and you want to have some quick reference to go to, the circle diagram is very easy to use. And I'll show you different uses for it today. Once we get done with all the basics today, I'll probably do most of the patho-phys Thursday. So that'll pretty much take up this week, is just working on cardio. Next week, we're going to continue with cardio, but focus a little bit more on the vessels. Again, we already talked about capillaries. We're going to do that again. We'll talk about properties of arteries and veins, because one thing that we try to do with the cardiovascular system is we try to regulate blood flow, always going from the high pressure end, which is usually going to start highest at the left ventricle, eventually back down to the lowest pressure end, which would be in the right atrium. So, you know, how we regulate that through various inputs of vessel tone and just going down a pressure gradient. We'll kind of talk about that. And then we will end our material for lecture 2 with the respiratory system and gas exchange. So all the material that's being covered for lecture 2 exam is just right in these two modules. So cardio phys, that's just one PowerPoint. We're going to do part of it today. We'll do part of it Thursday. We'll get through blood flow and pressure next Tuesday. We'll finish up with respiratory system and gas exchange next Thursday. The following week, we don't meet on Tuesday, so there's no class meeting for the 4th. For the 6th, this will be our Q&A session. So if we have anything that we didn't finish, we can finish it up then, or if we have any last minute questions, we'll use that. And that leads us into lecture 2. So again, I did kind of pull out the blood immunity and lab dye. We're going to do that as a separate exam. So I'll mention that again before we get there. But for right now, all we're really focused on is either finishing up lecture 1 and starting to work on cardio phys, but most of us we're working on lecture 2 now. Okay, so I wanted to kind of save or hold off on opening these modules. So if you haven't done your lecture 1 exam, you know, make sure you don't click on this thing and hit start, because now this is open. So you can go to quizzes as well, where you'll be able to see lecture 1 versus lecture 2 independently. But again, since I've already opened up the next modules, if you haven't taken lecture 1 exam, just be careful where you click, right? So don't click on that one. Make sure you go back to that one. But anywho, let me go ahead and open this up here. I'm going to do a lot of initial information on the page here, on the not on the PowerPoint, but on the paper. Let me go ahead and get it open. So we have it. And one of the first things we're going to do is this. We're going to do our little circle diagram. We're going to talk about blood flow through the heart. And then I'm going to talk about, in this particular order, we're going to do systole versus diastole. We're going to do various different types of input. You know, like what does the heart need to know in order to auto-regulate appropriately, or what does the brain need to know, we should say, in order to auto-regulate the heart appropriately. So input, output, once we understand what the body needs and we don't want to overpressurize it, how do we speed the heart up or slow the heart down? So we'll talk about output. We'll talk about various different types of cardiac output. So cardiac output equals stroke volume times heart rate. Cardiac output equals metabolic demand. Cardiac output equals venous return. We'll talk about coronary circulation of the heart itself, conduction system, and then we'll go back to this and we'll kind of show you how the heart normally fills up during systole and diastole. And we can talk about other factors that get involved that could lead to maybe heart changes. And then that'll be a good segue into Thursday when we actually talk about cardiomegaly, congestive heart failures, heart attacks, all that kind of stuff. So we're going to do a lot of basics, kind of starting with this diagram first. But the PowerPoint, it's set up in a way where it's kind of almost telling a story. It's basically saying, here's our heart, here's some things that can go wrong with it. And it does give you like a very generalistic scenario. Like sometimes a person might have damage to the heart wall, and that's usually due to some type of myocardial infarction. And then that can lead to other heart problems, like that could possibly lead to congestive heart failure. And when people have congestive heart failure, usually cardiomegaly is present. But it doesn't have to go that way. It's just, here's a commonality that we see in a lot of people with heart disease. And of course, heart disease itself usually has something to do with poor coronary circulation. But again, the test questions, they do come from the PowerPoint. So usually I'm just going through the PowerPoint saying, how can this be converted into a question? But today's lecture is going to kind of skip around a lot. So we're going to start by doing this, and we're going to do it on some paper. So I've been watching a lot of Bob Ross lately. That's what Oscar's watching right now. So let's see what we can do. Let's first draw a little happy circle. And inside our circle, we are going to subdivide it in half. All right. So again, if you had me for anatomy 1 and anatomy 2, this is something that we just drew again and again and again. If you didn't have me, just draw it. Just draw it with us, because this will be your go-to diagram for everything. So when we talk about systole and diastole in a minute, we can talk about this. When we talk about murmurs, we can use this. When we talk about congestive heart failure, this comes in real handy. So it's just one of those really useful diagrams. The whole point of it is to oversimplify circulation through the heart. So if I'm trying to trace a drop of blood, this is what this is primarily used for, but you can basically use it for everything. So when we draw the picture of the heart, heart's going to be facing towards you. So this is going to be our right side. This is going to be our left side. And that's pretty typical of anatomical drawings, because you're always kind of assuming that you're looking at a patient on a table laying face up or supine. So right and left switch over, but that's important to remember, even though this is your left hand and this is your right hand, the heart's facing towards you as if the patient is supine. So it switches. So that's right and that's left. I used to get that all kinds of messed up when I had to deal with patients, and like sometimes the patient would be prone, sometimes the patient would be supine, and you're trying to describe something right or left, and you're holding your hands out just to figure out what right and left are. It happens. So I'm going to draw something that looks like that. This is basically just representing an open valve. It doesn't really matter which position it's in, but that's going to be one of our valves right there, or one set of valves. Down here, I'm going to have another set of valves, even though I've already drawn the circle kind of closed. We're just going to draw it like this anyway. And I'll label these in just a minute. But the first thing we did is we just subdivided the heart into two halves, because essentially you have two pumps in one. You have what's called the pulmonic circuit, which is the right side of the heart, and then you have what's called the systemic circuit, which is the left side of the heart. So essentially what's going to be happening is blood's going to be coming back from the body. It's already circulated, and it's going to enter into this right side as deoxygenated blood that's high in CO2. So we need to get that to the lungs. So that's why we call this the pulmonary circuit or the pulmonic circuit. Once we get that blood to the lungs, though, gas exchange will take place. We're going to breathe off the CO2. Fresh O2 is going to enter. It's going to attach to the hemoglobin of blood. And then pulmonary veins is going to bring blood back to the left side of the heart. And the left side of the heart, which is the larger of the two sides, is going to end up pumping blood out into systemic circulation. So that's why this is called the systemic circuit. And I'll label that in a minute. But that's the gist. And again, where I'm placing the valves isn't hugely important. It's just trying to show easy flow of like a single drop of blood. Like if I'm trying to trace a drop of blood, then it's just easier if I put this valve down here rather than right there, right? Because then it's not going like this. So we're just going to put this valve here, this valve there. These are AV valves. These are semilunar valves. But again, this in reality would be right here. This one in reality would be right there, but that makes it harder to draw. So that's why we're doing circle diagram, because if you can trace a drop of blood in words, you know, where does it enter first? Where is it coming from? Where does it go through? Right? Where is it entering next? Where is it coming from? Where does it go to? If you can do it in words, then it doesn't matter what it looks like, because then once you look at the real picture, it becomes drastically easier. So circle diagram, right side, left side, top two chambers, atria. So right atrium, left atrium. I will, as we're drawing this, let me put some of the key terms that we're going to be using up here on this separate piece of paper, because it's going to get real messy real fast. So atrium is the A. So right atrium and left atrium. So atrium or atria, singular plural. Think of these as receiving chambers. That's what I want you to think about. Think of these as receiving chambers. They're receiving some type of venous return, okay? So entering into the right atrium, we're going to have the venous return of the whole body, basically. Entering into the left atrium, we're going to have pulmonary veins. So venous blood is going to be entering into atria. So we're going to think about these as receiving chambers, okay? So the top two chambers are atria. Bottom two chambers are ventricles, and the ventricles will be our pumping chambers. So I'll label this one right here, and I'll label this one right here, okay? So ventricles, it's Oscar in the background. Ventricles are going to be, whoops, sorry, I meant to put left ventricle and right ventricle. These are going to be pumping chambers. Okay, so think about atria as receiving, think about ventricles as pumping, okay? And it doesn't mean the atria can't pump, they can, but they're just not great pumps. They're very small. They're not as thick and strong as ventricles are. And this will just help you visualize things later. So you got the two atria up top, the two ventricles down at the bottom. These are receiving blood from somewhere. These are going to be pumping blood to somewhere, okay? Separating your atria from your ventricles are going to be what's called AV valves, right? So these two valves are going to be our AV valves, right? Standing for atrioventricular valves. So atrioventricular valves, there's two of them. You have tricuspid and bicuspid can also be called mitral, okay? So this is our tricuspid valve on the right. This is our bicuspid valve or mitral valve on the left. How do you remember which valve goes with which side? Just use the mnemonic lab ret to remind you, okay? So if it's the left side, it's bicuspid. If it's the right side, it's tricuspid. Okay? And then you just have to memorize that the bicuspid is also called mitral. So you call it a lamb rat if you want to. Easy peasy. And then down here, these two valves are going to be our semilunar valves. So semilunar valves are what's separating the ventricles from their destinations. So those are going to end up being your pulmonic valve and your aortic valve. So this is going to be our pulmonic valve right here. This is going to be our aortic valve right here, which I'm going to write out so it's not confusing. Because earlier we already used the abbreviation AV to stand for atrioventricular valves, which that's what we would call bicuspid and tricuspid together. Like if I'm trying to describe these two at the same time, what are they? They're valves that separate the atria from the ventricles. So that way we don't get backflow when the ventricles contract, right? So together they're called atrioventricular valves, separating the atria from the ventricles. The pulmonic and aortic valve together are called semilunar valves. That's what separates the ventricles from their destinations. So if we're tracing blood that's coming through the heart, a lot of times they want you to name every chamber, every valve that you would have to pass through, similar to like tracing food through the digestive system. So if there's a sphincter I got to go through, make sure you name that too. If there's valves we got to go through, we need to name those too. And it doesn't matter where you start, but a lot of times we'll start on the right side because we're going to start off with deoxygenated blood entering into the right atrium. So this deoxygenated blood is obviously low in O2, but at the same time, it's high in CO2. And we'll talk about numbers with this when we get into respiratory system and gas exchange. So we'll get into the partial pressure of oxygen and the partial pressure of carbon dioxide. What does it mean to be low or high? How would you read that in a patient? But for right now, this is old blood that's already circulated around. So that's why it's already dumped off some of the O2, right? It picked up CO2 as it was going through circulation, and it's now coming back to the heart to exchange these gases. So this was basically venous return of the body, which we usually just abbreviate that VR, right? So VR is just going to basically stand for venous return of the body. And this is implied. So venous return of the body. And when I'm draining different parts of the body, veins go through a process called consolidation, where you start off with many, the more distal you are, and they become fewer as they go back toward the heart. So if I'm draining head, neck, and arms, that all eventually dumps into a superior vena cava. If I'm draining the entire lower part of the body, which is basically everything beneath the heart, that's all going to dump into an inferior vena cava. And if I'm draining the heart itself, then that's going to drain through a coronary sinus. So I'll write all that out again. So VR is going to be made up of superior vena cava. So I'm just going to abbreviate this: superior vena cava, inferior vena cava, and coronary sinus. This drains head, neck, and arms. This drains all the lower body, which basically would include the entire digestive system, everything that's coming from the lower extremities, everything below the heart, basically. And then the coronary sinus is draining the heart itself. So this is venous drainage coming from the whole body, so we just call it the VR, right? And again, it's old blood, so it's low in O2 and high in CO2. So where do we want to take that? To fix that, we want to take it to the lungs. But we're going to move from our receiving chamber, which is the right atrium, to our pumping chamber, which is the right ventricle. So to do that, we need to pass through an AV valve, and on the right side, we call that the tricuspid valve. So we're going to go into here. And eventually this thing's going to fill up, mostly passively at first. We'll talk about numbers and percentages later. But premature closure of the tricuspid valve is going to lead to what's called atrial systole, which is going to then fill up the right ventricle. The heart's going to then squeeze on that, slam the tricuspid valve shut, force the pulmonic valve open, and then blood will leave that right ventricle and go toward the lungs. But we have to pass through some structures to do that. Like the first structure that we enter once we go through the pulmonic valve is going to be the pulmonary trunk. And I'm just going to go ahead and write this out since we got room down here. And the pulmonary trunk let's pretend, again, we're not trying to draw something anatomically correct here. So let me just put a little disclaimer on here. This is not anatomically correct. Okay. Clearly, but your pulmonary trunk is the first major vessel that comes off the most anterior portion of the heart. So coming up above the heart, this is actually called your base of the heart, and that's where the major vessels come off. It's kind of stupid, I know, but this is the base, this is the apex. It's just harder to draw that when you're drawing the real deal, okay? But it would be something like that. So here's your pulmonary trunk, and that's going to subdivide into right and left pulmonary arteries, which then at the same time that's happening, your trachea is also splitting and forming a right principal bronchus and a left principal bronchus, right? And they'll follow each other through the lungs. So as these pulmonary arteries, which always carry blood away from the heart. So this is the right pulmonary artery. This is the left pulmonary artery, right? Arteries always carry blood away from the heart, right? So these are pulmonary arteries. And what's always going to occur in arteries always carry blood away from the heart. Okay. So this is going to be deoxygenated blood. So in a picture, it's going to appear blue. Again, it's going away from the heart. So that's an artery. So your pulmonary trunk is an artery, right? And that's going to then carry blood into the bifurcated vessels, which we just simply call pulmonary arteries. I'll write that out. Normally, you can just abbreviate arteries AA. And that will, of course, keep dividing eventually until we get down to capillaries and alveoli. And when those two meet, that's where gas exchange is going to take place. So we'll talk about that more when we get into the respiratory system. So while we're in the lungs, gas exchange is taking place. The CO2 is going to be breathed off. Fresh O2 is going to be entering, right? And to get that back to the heart again, we must travel through pulmonary veins. So PVV is going to equal pulmonary veins. That's an O. Right? Pulmonary veins. So this is coming from the lungs, obviously. And pulmonary veins are going to bring blood back into what? A receiving chamber, so an atria. Atria are always receiving blood from some type of venous return. And this is now fresh blood, so it's high in O2, low in CO2. And we want to pump that fresh blood back out to the body again. So we need to go through an AV valve. In this case, it's the bicuspid or the mitral valve. And we're going to fill up that ventricle. Eventually, it'll get filled up. Doesn't take much time, but it takes a couple of steps. And we're going to build up some pressure in here. So as the pressure builds up in here, eventually this valve is going to get slammed shut. This valve is going to get forced open, and blood's going to get ejected out, right, into systemic circulation, right? So this is basically going to the whole body, right? This is going back out to the body. So it's a complete circle of old blood from the body coming into the right side of the heart. It's deoxygenated blood, so it's low in O2, high in CO2, and we need to fix that. So that's the whole function of the right side of the heart, is to take that venous return that's coming back from the body. It's already been delivered, and now it's coming back and reoxygenate it. So that's what the lungs do. So the right side of the heart is referred to as your pulmonic circuit. Whoops, just circuit. While your left side of the heart is going to be your systemic circuit. Okay. So once we've exchanged the blood gases in the lungs and we have freshly oxygenated blood coming back in, now we want to take that oxygenated blood back out to the body again. So that's the job of the left side of the heart. So this is considered our systemic circuit, or it's responsible for systemic circulation. And of course, the aortic valve opens up into the aorta. And the aorta, it's the largest of all the vessels. It's the strongest of all the vessels. It's basically where almost all the rest of your vessels come from. Oh yeah, sorry, Courtney, I didn't see that. Yes. So if it's PAA on the PowerPoint, pulmonary arteries. Yeah. Let me type that in chat too. So sometimes in anatomy, AA or AA can equal arteries. BV or VV can equal veins. Welcome. You know, we don't usually do capillaries that way, but NN or N equals nerves. So, and so I imagine AA and VV are probably in the notes quite a few times. So that's what those stand for. It's just shorthand anatomy. It's the way Grey's Anatomy, the book, TV show, does it. And another thing about the aorta is if I want to deliver oxygenated blood to the body, this is where it has to come from. So if I'm the heart and I need to receive oxygenated blood, it's got to get it from the aorta. If I'm the lungs and I want to receive oxygenated blood, that also has to come from the aorta, right? So there's no exceptions to the rule. If you're receiving oxygenated blood, it was a branch off that aorta. So easy peasy. So one more time. Now that we know what all the terminology is, right side, left side, right atrium, left atrium, right ventricle, left ventricle, right? This is our AV valves right here. This is the tricuspid valve on the right. This is the bicuspid valve or the mitral valve on the left, right? Down here at the bottom, this is where we're going to put them. This is going to be my pulmonic valve. This is going to be my aortic valve. This leads into the aorta, which is essentially going to be the vessel that carries all oxygenated blood to the body. But how did we get it that way? Well, first of all, it started off as deoxygenated blood heading back to the heart through the venous return, right? And we're draining the head, neck, and arms through the superior vena cava, the lower body through the inferior vena cava, and the heart itself through the coronary sinus. That enters into the right atrium as low in O2, high in CO2 blood, okay? We want to take that to the lungs to get rid of that and to get more of that. So we have to go from the right atrium through the tricuspid valve into the right ventricle, through the pulmonic valve into the pulmonary trunk, and then that will lead into pulmonary arteries, right and left, and that goes to the lungs. So this is the pulmonic circuit. Once we get the blood to the lungs, now we're going to take it back to the heart. So from the lungs, we're going to head back to the heart through pulmonary veins, because veins always carry blood toward the heart, okay? And I know that light's flash. Let me turn on the overhead light so it doesn't keep doing that. A lot of times this little camera tries to like auto correct for the light. So hopefully if we have more light, I'll stop doing that in a minute. But veins are always going to carry blood toward the heart, okay? So that's the two things we remember. Arteries carry blood away from the heart, right? Veins carry blood toward the heart. Just remember that. Don't worry about oxygenated versus deoxygenated because it gets switched in pulmonic circulation. So as we're going back toward the heart through pulmonary veins, that's going to enter into the left atrium. We're then going to pass through the bicuspid or the mitral valve to get to the left ventricle. Left ventricle is the biggest, strongest part of the heart. That's what's going to overcome what's called peripheral resistance, so we can push that blood into the aorta, right? And the aorta is going to deliver that blood to all parts of the body. Through capillaries, of course, the gas exchange and the nutrient exchange will take place. And then from the capillaries, the venous system will form, right? And then the veins will bring the blood back to the heart again, and you just keep repeating this cycle. So that is the gist of it. And again, this is assuming that everything's working appropriately. So. There you go. So it's a very quick rendition that you can recreate in just seconds. And that's what's nice about it, because that's a lot easier to draw than something that looks like that. And that's not even a very detailed diagram. You know, it gets way more detailed. I don't know if I have pictures of it or not. Probably not. You know, something more like that. It's hard to draw, right? So it's hard to visualize if you can't draw it. And if you're trying to figure out, well, what's happening if I have some type of murmur, you know, it's easier if you can see those things. You know, what position should a valve be in during different phases of the heart cycle? So that's why we want something simplistic to draw, because if you can visualize what's happening in the circle diagram, you can then apply that to the more complex diagram. So that was step numero uno. And if you can master that diagram and the capillary diagram, I promise you, you can be a cardiologist. You just watch a couple of YouTube videos on EKGs, you're good to go. Practice it for about a month or two, you'll get it. But yeah. Next thing we're going to talk about is systole versus diastole. So systole, this is what I want you to think about. Systole is contraction phase of the heart. Everybody knows that, I'm sure. During the contraction phase of the heart, what we're trying to do is overcome peripheral resistance. So we kind of have to squeeze on the blood. Heart's basically a hollow pump. It's a couple of pumps, you know, it's two pumps in one. So if we're focusing on what's going on in the left ventricle, that's usually what you're visualizing during systole and diastole. We're trying to eventually overcome the peripheral resistance of the body, which there's a lot of factors that go into peripheral resistance. You know, if I'm hypertensive under stress, then there's more tone we have to overcome. If I'm a bigger person, there's more distance that the blood has to travel. All those can increase peripheral resistance, and the heart has to overcome that to get blood out of the heart into the arteries. So the contraction phase of the heart is meant to overcome that peripheral resistance. But we have to have blood to work with, right? You can't really contract on nothing. It wouldn't generate much pressure, at least you could. So during diastole, this is what I usually start with, actually. I usually always do diastole first, and then I do systole. Think about this as the time where you're refilling the heart. It's the refilling phase of the heart. Don't think about relaxation, because that doesn't tell you anything. Relaxation is a worthless word. So we're going to use contraction versus refilling. And the trick that you sometimes run into with this, and it's not always in a book, is when the words systole and diastole are being mentioned alone, that implies ventricular systole and diastole. So if all I see is the word systole, then it means ventricular systole. If all I see is the word diastole, then it means ventricular diastole, right? But if I'm talking about atrial systole, then that has to be included. Like I would have to put the word atrial there to include that. And there's a good reason for that. Hopefully we don't need to get that complicated. But by themselves, if I just see the word systole and diastole, it's just referring to ventricular systole and ventricular diastole. Okay? So with that being said, if I were to draw a heart in diastole and ask myself, well, what valves need to be open and what valves need to be closed to refill the heart? What are we trying to refill? Trying to refill the ventricles, right? Because it just says diastole. So as blood's coming in to the atria, and then from the atria to the ventricles, what valves should be open? So during diastole, AV valves should be open. While we're trying to build up pressure, can't just have it leaking right back out again, right? Semilunar valves should be shut. Okay, because we're trying to refill the ventricles. Okay, during systole, I'm now taking these ventricles that have been filled, and I want this blood to leave, right? So during systole, or the contraction phase of the heart, it's talking about the ventricles contracting and back pressure, because when you compress this blood in here in this chamber, the blood's going to push against all the walls, right? So when you compress the blood in the chamber, it's going to start pushing against all the walls, and that's going to slam these AV valves shut. We'll get more specifically into the timing of that in just a little bit. It'll force the semilunar valves open, right? But we needed to overcome pressure in here first. So we had to build up some pressure in here first, but now the heart can eject blood out, right? So during systole, what valves need to be open, what valves need to be shut? Well, we don't want blood backing up, right? So AV valves need to be shut. And then the semilunar valves, of course, are going to be forced open. And during the shutting of that AV valve, that's what creates that first heart sound, right? When you're starting to build up pressure inside of here and the heart contracts on that volume of blood, it slams that AV valve shut. And so it goes, and that's what gives you that first lub heart sound. So S1 is the closing of that AV valve right at the beginning of systole. Once the blood leaves, right, pressure inside here is going to drop drastically. Peripheral resistance will then be higher than intraventricular pressure. So the pressure out here will eventually be greater than the pressure in here once the blood leaves, right? And then the back pressure will shut the semilunar valves, and that gives you your second heart sound. So simple use of the circle diagram already. Okay. So systole versus diastole, and we'll come back to that toward the end, because we want to talk about the role of what's called atrial systole. Next thing we're going to talk about is some physiology, control mechanisms of the heart. Most of your heart control is coming from the medulla oblongata of the brain. If you ever get asked about any nuclei within the brain that have to deal with heart control, there is something called nucleus tractus solitarius inside the medulla that plays a crucial role in being able to regulate heart action. It's receiving a lot of the information that we need to control the heart. And the type of input that the medulla needs in order to properly regulate the heart is going to come in the form of pressure and chemical changes. So baroreceptors are baroreceptors and chemoreceptors are chemoreceptors. These are bringing in information to the medulla so the medulla knows what's going on in the body, right? Like when we're trying to control the heart, I don't want to overpressurize the artery side. I don't want to overpressurize the venous side. So if I have increased artery pressure in the arteries, I'll just put increased artery pressure. That might indicate slow heart down. Because if I got too much pressure going into the aorta, then that's going to equate to too much pressure going into the coronary arteries, which is going to indicate too much pressure going to the brain. And we don't want to overpressurize stuff because that could lead to aneurysms and all kinds of problems, right? A dissecting aneurysm, which could lead to a stroke and no good. So that might be an indicator that we need to slow the heart down. But if I have too much pressure in the veins, too much venous pressure, that might mean that we need to increase heart activity, right? So that's just an example. That's just an example. With chemoreceptors, the more important of the two, believe it or not, this is what's going to be the main control mechanism of your heart, right? Obviously, again, we don't want to overpressurize. That's no bueno. So we do have reflex phenomenon. Like usually once your coronary arteries get too pressurized, that'll send a vagal signal to the heart to immediately slow it down. So we'll talk about that later. But chemoreceptors equals your primary mechanism for controlling the heart, right? So what's the primary mechanism for controlling the heart? CO2, and we can just go ahead and put in hydrogen ions as well. Okay. And this is your main type of information that the heart needs through the medulla. Obviously, the medulla is what's reading it, but it's the main control mechanism for the heart, right? So medulla needs to know what are the CO2 and hydrogen ion levels in the body, because this equals how much metabolism is happening. Sorry for the noise in the background. It's city truck picking up branches or something. So a lot of times this ends up shocking most people, because a lot of people think O2 is going to be the more important chemical, right? Because what's the heart trying to do? It's trying to carry oxygenated blood to the body, right? And we do have receptors in your aorta and your carotid arteries that specifically pick up O2. But those are what's called peripheral chemoreceptors. Central chemoreceptors in the brain are only concerned about CO2 and hydrogen ion, because this tells me two things. This only tells me one, right? If I'm generating a lot of CO2, that means O2 has already been spent. So it tells me how much O2 has been used. Plus this contribute to pH changes in the body. So this is two versions of information. This is only one version of information. So this is your primary control mechanism. And once we receive input about pressure and chemical changes in the body, then, when we're doing output, how is the heart generating a basal rate? So basal rate of heart control is kind of like, what's your baseline? Like, what would it run at if we had no input whatsoever? And a lot of the basal rate of metabolism of the heart is controlled by fluid electrolyte balance, fluid/electrolytes. So blood levels of sodium, calcium, and potassium. So if your fluid electrolyte balance is all messed up and you're hyper or hyponatremic, calcemic, or kalemic, that's no good for the heart, because that's what controls its basal rate of metabolism. And your heart is what's called self-excitatory, right? So it's essentially the influx of sodium, like let's pretend this is a cardiomyocyte that's an excitatory cardiomyocyte. So an excitatory cardiomyocyte is like a specialized heart cell that's going to generate action potential, and that action potential is going to fire across heart cells, because cardiac muscle cells are interconnected. So if I can generate a signal here, it can flow across the whole heart, and that ends up forming what's called a syncytium, or sometimes people call it a syncytium. Right? So you can throw a Y in there if you want to, you don't have to. Some people call it a syncytium, some people call it a syncytium. But these cardiomyocytes, similar to neurons and other muscle cells, need to be depolarized. And the depolarization of cardiomyocytes usually starts with a little bit of influx of sodium, and that'll take you a little bit of the way. And then these sodium channels will kind of close and calcium channels will open. And then calcium takes you most of the way to depolarization. Okay. And at the same time, potassium's leaving during this, which will also, change the conductivity of the cell membrane. But that's what's going to generate our signals in the heart, is this kind of opening of sodium channels, opening of calcium channels, right? Again and again and again. So that's what's going to cause the cardiomyocytes to fire. That's your basal rate of metabolism. And in normal people, that'll occur a certain number of times per minute. So maybe 100 beats per minute is what that would normally run at with normal fluid electrolyte balance, okay? But that's weird, isn't it? Because if we were to test somebody at rest right now, what is normal resting beats per minute, it's usually about 72, right? So, I mean, that's just an average number. Anywhere from 60 to 100 is normal, but about 72 beats per minute is normal. So how are we going from 100, which is the basal rate of depolarization or the basal rate of metabolism for the heart, to 72? Well, there's two things I can do to modulate the heart. So heart modulation is altering of that basal rate. Heart modulation alters the basal rate. It either makes it more easily depolarizable or harder to depolarize, alters the basal rate. So if I want to speed anything up, we can use sympathetic. Sympathetic increases the basal rate. If I want to slow anything down, we use parasympathetic. That slows down the basal rate of metabolism. So if my normal resting heart rate is 72 beats per minute, but my basal rate of metabolism has me at 100 beats per minute, that means at rest, your heart is receiving at least some parasympathetic innervation, right? So acetylcholine. So when I'm sending signals to the heart through the vagus nerves, right? Through the vagus nerves, cranial nerve 10, that's going to slow the heart rate down. Okay. But if the heart is receiving norepinephrine, and you could even add epinephrine to it, right? Then that's going to increase things like heart rate and heart contractility. Okay. So when we're modulating the heart, so heart modulation, it's altering that basal rate of metabolism to the proper level that it needs to be based off input, right? So if you just take all the brakes off the heart and you don't accelerate it at all, it's going to fire at 100 beats per minute. So if I just were to clip every single neuron that went to the heart completely, it would fire at a normal rate of 100 beats per minute. But based off the information that your medulla is receiving about pressure and blood carbon dioxide and blood oxygen saturation, or the partial pressures of those things, it properly modulates it, right? So it says, I need you to go faster because we're producing CO2 too fast and we need to get rid of it faster. Or we're not producing CO2 quite that fast. Let's go ahead and slow down a little bit. So parasympathetics is going to equal the brakes, and sympathetic is going to equal the accelerator. Easy, right? Easy peasy. So again, some basic information, but it can be applied in semi-complicated ways later. Like one thing that we might mention a little bit later is what if a patient's low in O2, but CO2 is not high either. And the reason that they're low in O2 is because they have carbon monoxide poisoning. Well, we need to give them more O2, right? But because the body doesn't respond to oxygen nearly as well as CO2, not only are you going to give them O2, but you're going to give them CO2 as well, right? Because we need to increase that heart rate. We need to increase the respiratory rate so the oxygen that we're trying to give the patient can go to the body better, right? So that's when, knowing the basics helps, even when it gets to be more complicated and things aren't working appropriately. Next, okay, cardiac output equals stroke volume times heart rate. Cardiac output equals metabolic demand, and cardiac output equals venous return. Okay. This is what we might call a quantitative description of heart action right here. This is called a qualitative description of heart action. And then this is a limiting, or it could even be a stimulating factor, right? So this, we'll just call this a factor. Right? And this by name is called the Frank-Starling law. I like law. or you can call it the Frank-Starling mechanism. You can use it to your advantage. So when we are trying to modulate the heart appropriately, we're trying to modulate cardiac output. And cardiac output is the amount of blood ejected, if you can say, to the body, pushed by heart to body per minute. Okay. So it's cardiac output's the amount of blood that we're pushing by the heart to the body per minute, right? And we can measure that by combining the two factors of stroke volume times heart rate. So normal, if I am, say, 160, 170 pound, I'm pretty sure that's probably off, or a 70 kilogram male, then my stroke volume is going to be about 70 milliliters. Okay, so about a milliliter per kilogram. My normal resting heart rate is about 72 beats per minute. And when you multiply those two together, that gives you about 5,040 milliliters, which is basically the equivalent of 5 liters. Okay, so when they ask you what's normal cardiac output, about 5 liters. That's where that number comes from. And it's based off a 70 kilogram male who has a resting heart rate of 72 beats per minute, and you're pumping out about a milliliter per kilogram. So if I'm a 100 kilogram male, then it would be 7 liters per minute. But anywho, this is the normal number you would give. It's based off that calculation. So again, it doesn't satisfy everybody. It's just a normal average value. Easy, right? So when you get into this number, what would happen if my body is under stress, right? If I'm under stress, my cells need more metabolic energy, right? So sympathetics is going to kick in, and stress will increase sympathetic tone in the body, we sometimes call that. And increased sympathetics is going to increase my cardiac output. And when I increase cardiac output, it's because I'm altering these two things. So how can I alter cardiac output? Well, I need to either alter stroke volume, or I need to alter heart rate, or both, right? But these are the determining factors of what cardiac output can be, right? So if I need to pump out 6 liters per minute, again, I could just increase the stroke volume by itself, remain at the same heart rate. I could increase the heart rate by itself, remain at the same stroke volume, or I could just increase both, right? So normally what ends up happening is norepinephrine by itself can increase heart rate. You add epinephrine to the mix, or adrenaline, and that'll usually increase stroke volume. So sympathetics, of course, can increase cardiac output pretty immensely, because we can alter both heart rate and stroke volume. Parasympathetics usually just alters heart rate. It doesn't really do much to stroke volume. It just goes back to normal. Whatever the normal resting basal metabolic stroke volume was, that's what it remains, but it can slow it down. Okay. So, and we'll talk about, what are some things that we can do to, again, alter stroke volume. Let's go ahead and talk about it now. So factors that affect, let's say, stroke volume. One thing is maybe how strong the heart is, right? Is the heart strong enough to compress blood well? What about the peripheral resistance? Is the peripheral resistance too high? Right? Where if I have a weakened heart, am I able to overcome that peripheral resistance? But a lot of it has to do with contractility. So factors that affect stroke volume usually have to do with your contractile capability. And it's how vigorously the heart can squeeze the blood, right? So for example, and there's examples in your notes that'll go over this. If I just increase contractility alone, let's imagine that this is a left ventricle. So let's just focus on what's happening inside of here, right? Inside of here. A normal left ventricle. So again, we're just focusing on left ventricle. A normal left ventricle can hold about 135 milliliters of blood, okay? But during rest, I don't need to pump all that blood out at once, right? We can pump more under times of stress, but at rest, if I have 135 milliliters of blood in here, and let's say I pump out 70 milliliters, that's due to the heart beating something like this, okay? So imagine this is the left ventricle in between my hands, right? And this is the amount of blood that would fill that ventricle, okay? So if I squeeze my hands this much, some of that blood's going to leave, but some of the blood's going to stay because I haven't completely forced all the blood out, right? I'm just doing this, right? So that would be the heart beating at rest. So if I had 135 milliliters in there, so let's say that this is 135 milliliters total, right? And I compress it like that, 70 milliliters will leave and 65 milliliters stays behind, okay? So 70 will leave, 70 milliliters leave, 65 stays behind. But what if I compressed it like this? Right? Now, 135 milliliters is leaving, 0 milliliters is left behind. So pretty simple, right? Yeah, no problem. So again, right now, we were diving a little bit deeper into cardiac output equals stroke volume times heart rate. And in a minute, we'll talk about cardiac output equals metabolic demand. We'll talk about cardiac output equals venous return. But let's just start with the quantitative description of cardiac output, which is kind of going over how much the heart beats per minute, like how much blood does the heart push out per minute. And at rest, it's normally about 5 liters, right? And what goes into determining what that cardiac output needs to be, of course, is the medulla receiving signals from baroreceptors and chemoreceptors. And at rest, we're just not generating a ton of CO2, right? So the signals aren't going to be that strong that we need to increase cardiac output. So let's go ahead and slow down the basal metabolic rate. Let's go ahead and beat about 72 beats per minute because we don't need to remove that much CO2. Let's keep stroke volume kind of normal. And under normal circumstances, the heart doesn't super vigorously contract. Like if I'm sitting here at my desk right now, I don't feel my heart pounding, right? Because it's not, right? It's just doing like this. Right? So that's part of what affects stroke volume, is just how vigorously the heart is compressing, right? So one way that I can increase stroke volume is by simply contracting more on a normal chamber, okay? So if a normal chamber holds up to 135 milliliters in under resting conditions, only about 70 of those leave, about half of it leaves. But under a more stressful situation, if I add more epinephrine, then more will leave, okay? But believe it or not, we can actually change how much blood will be held in that left ventricle by preloading it a certain degree. And I'll explain what preloading means, okay? So one thing that I mentioned is a lot of times when we're focusing on, like say, systole and contraction, we're focusing more on the ventricles. We'll come back to the atrium, we'll talk about those later. But it's the ventricles that are your big pumps. And your main pump is your left ventricle. So a lot of numbers and information that you read about is coming from pressures and volumes that the left ventricle has to deal with. So that's where we're going to focus, right? So if my heart, under normal circumstances, holds about 135 milliliters in here, and that's at rest, and normally 70 goes out, it's pretty easy to see how we could increase that stroke volume, which is how much we're pushing out per beat. So stroke volume equals the amount of blood ejected per beat. That's another way to say it if you want to, okay? It's talking about what's leaving that left ventricle, right? And normally whatever leaves the left ventricle, let's say 70 milliliters, heads out to the body. If nothing else changes in between, 70 milliliters is going to get pushed back in, right? And that 65 was left in here, right? 70 came back in. And now you're back up to 135 again. Everything's hunky-dory and everything remains kind of at rest. But there's another way I can increase stroke volume other than just by increasing contractility. Another way I can increase stroke volume. So one way we can increase stroke volume is through contractility. Like how hard is the ventricle beating? But another way I could increase stroke volume if I want to is to simply preload that left ventricle more, right? And when you're preloading, you're talking about distension of the ventricle. So again, let's imagine that here's my left ventricle and it's holding 135 milliliters like this, okay? And when I contract, 70 left, 65 remains behind, 70 comes back in again, I go right back to 135, right? So if I had 135 to begin with, let's just put it in a cup maybe. All right, I have 135 in here total, 70 left, right? 65 stayed behind. And I refilled this cup with 70 more milliliters, I'm right back to 135 again though. But what I'm telling you now is this isn't a fixed volume like a cup is. You know, this thing's made out of glass. This is made out of muscle tissue, which has some distensibility. So I can actually change how much volume can actually fit into the left ventricle by altering its preload, okay? So let's say that during one beat, 70 milliliters left, 65 milliliters stayed behind, but during diastole between beats, 90 milliliters came back in to fill this thing back up again, okay? So now I still had 65 left in here, 90 comes back in, right? How much am I holding now? Now at 155. So the heart normally only holds 135, right? So what's happening to the heart? It's getting distended a little bit, so it's getting stretched out. And that natural snapback, like if you stretch a rubber band this far, it's going to snap back that far. But if I stretch a rubber band this far, it's going to have more, you know, snapping power. Same thing with the heart. If my heart normally only stretches out this far to hold its normal 135, right? It's going to have contractile power because it's a muscle. But if I stretch that a little bit more ahead of time, it's going to have a little bit more contractile power because I've now distended it a little bit. And that's called preloading the heart. So when you preload the heart, it's actually causing some distension because you're adding more fluid to it than what it normally holds. And so that stretches it out a little bit. It's going to have that natural snap back to it, right? So in this case, if I preloaded it by adding 90 milliliters and I didn't change anything about contractility, if it just snaps back and contracts normally, then my output's going to be 90 milliliters, right? So the next output would be 90 milliliters again if I didn't touch contractility. That's the Frank-Starling law, or the Frank-Starling mechanism that says cardiac output equals venous return. Because your heart can be distended or stretch out, it's called preloading it. And by preloading it, we're distending it a certain amount and it's going to naturally want to snap back. But now I'm telling you, not only can you increase contractility, not only could you preload the heart, but you can do both, right? So what we've been talking about so far are factors that can affect cardiac output. And those factors are stroke volume times heart rate. This one's pretty straightforward. I can either increase beats per minute or I can decrease beats per minute and leave the stroke volume the same. Either way, that's going to increase or decrease cardiac output, okay? But increasing rate, which is the simple one to understand, is not the only way we can alter cardiac output. I can also increase stroke volume, right? And one way I can increase stroke volume is just to increase contractility, or we sometimes call that heart action. They sometimes call that heart action. But I could also increase preload by distending the heart more, or I could do both. So now imagine what we were doing before. Imagine what we were doing before. You had a heart, and again, let's just focus on left ventricle. Let's take all the rest of it out. Who cares? So in the left ventricle, this thing normally held 135 milliliters. Under resting conditions, let's kick out 70 milliliters. Okay. And so we haven't changed any contractility. That's a 7. Right. We haven't preloaded it any. And then normally what would happen is 70 left, 70 usually comes back in, because whatever we're pushing out, that's going to push the venous system that much further forward, right? And the venous system will dump back in. Whatever I push out normally gets dumped back in, but I can make alterations in between, right? I can change what the heart's pushing out, which is going to push in more. I could even change vessel tone out here, and that can change this without affecting that, but it will. So again, let's keep it as simple as we can first. So if I have 135 milliliters in here to begin with, under normal contractility, we don't compress it all out, right? So I push out 70, 65 stays behind. Okay. But one way I could increase stroke volume is just simply to make the heart more contractile, right? I can squeeze it harder, which would increase my ejection fraction. So maybe I'm ejecting 90% instead of 80%, or maybe I'm compressing out 100% rather than 60%, something like that, right? But contractility can change your stroke volume, right? This is stroke volume right here. So I can increase that by increasing contractility, or I can increase this by preloading it more. And one way we can increase preload, like how could I make that number go up? Like how could I make that number go up? So preloading is amount of distension. It's the amount of stretch that the left ventricle is having to go through. And if I want to increase the preload, increasing the stretch of the ventricles, usually I'm going to increase my venous return, right? And we can do that through sympathetic innervation, perhaps. Like if I send more, let's say this is a vein and there's blood in it, veins are naturally very distensible. But if I just contract it more, because we have circular muscles around it, by sending, say, more norepinephrine or more angiotensin II or more vasopressin, right? Some of the vasoconstrictors of the body. So vasopressin is the same as, say, an antidiuretic hormone. If I send any of those to the vein, that's going to cause the vein to compress more, send more blood back to the heart. When I send more blood back to the heart, let's say 135 was in here, 65 left, or sorry, 70 left, 65 stayed behind, but now we're sending 90 milliliters back in. Now we've just distended the heart a little bit, okay? And so now I'm at 155. And if I just contracted at normal rate, right, which was this, I would send out the same amount that just came in, because not only am I contracting, but I'm also pushing back from that amount of distension. Okay, so heart like this. If I'm preloading it, it's now like that. So it's distended. But I can also do both. I can have normal. I can have increased contractility. I can have preloading it, increasing distension. And then I can do both at the same time. I can increase distension, snap back, and increase contractility, forcing it all out. So now look at how much it's changed. We've almost doubled the amount of stroke volume. So that's why the heart is able to drastically change so much. Right? So at rest, I might be pushing out 5 liters per minute. When I'm doing vigorous activity, it might be 35 liters per minute. Huge difference, huge difference. Is that weird? Is that hard? So I know a lot of these, this is kind of going in a different order. When we used to do this in Anatomy 2, this would have been the Cardio 2. So if you were my students, this was Cardio 2 that we started talking about. Cardiac output equals stroke volume times heart rate, and cardiac output equals metabolic demand. So it does bounce around quite a bit. But I can show you in the notes as well where some of this information can be found. Let's see. So here's cardiac regulation. Sympathetic and parasympathetic, speed up, slow down heart, right? They receive their input from the medulla. Here's cardiac output equals stroke volume times heart rate. Here's the normal numbers. Here's cardiac output equals metabolic demand. This even gives you how you calculate stroke volume, which is kind of overly complicated for a very simple idea. Stroke volume is equal to end diastolic volume minus end systolic volume. So that's kind of what I was drawing for you a little bit, even though it didn't really look like that. End diastolic volume, just basically, I'll write it out. End diastolic volume is basically just how much fluid is in that, say, left ventricle at the end of diastole, right? During diastole, we're refilling the heart. So normally it's about 135 milliliters, okay? So at the end of diastole, once we've refilled your ventricles, how much is in there? About 135 milliliters, okay? And when you're trying to calculate stroke volume equals end diastolic volume minus end systolic volume. So what leaves, right, was 70 milliliters. What was left behind is 65 milliliters. So inside the ventricle, that's the end systolic volume. So after systole took place, how much was left in the left ventricle? 65 milliliters. So this ends up being the stroke volume. So this is the stroke volume. This is end diastolic volume. This is end systolic volume. So that's what I was showing you. And there's also a little kind of picture I made for you in here, because I know it's hard to follow sometimes. It has all these numbers in there too. So normally when we're doing stroke volume at rest, the contractility is not all that great. We're not really preloading the heart. And I even simplified the numbers for you. You know, let's just pretend it's 100 and 70 milliliters leaves. How much is left behind? 30, right? So you don't even have to use the more complicated numbers to understand the basic idea, okay? But ways that we can change stroke volume is I can distend the heart more. I can make the heart contract more vigorously, or I can do both. So it's not terrible. It's not terrible, but all this stuff's in here for you. I'm not going to leave you hanging. It's all there. If some of this is new, that's fine. It's okay. It's one of those better late than never type things. You know what I mean? And everything we learn is new, at least at one time. So we'll get there. Little by little, we'll get there. And this is one of those things where if you don't understand it, then ask me to go over it again. You know, if your stroke volume's not normal, it's indicative of maybe too much afterload and the heart's normal and just can't overcome it. Or the heart has changed and it's no longer capable of going against either normal peripheral resistance or the increased peripheral resistance that was already there. You know what I'm saying? So if we're not ejecting enough, maybe the heart itself is broken, right? Maybe the peripheral resistance has drastically changed for whatever reason. Like if I have a car sitting on top of my chest, my heart might be fine, but the peripheral resistance is just too great at that time. So my heart's not able to eject as much as it normally could, right? And sometimes people get vasospasms that can cause that, right? But another problem is my heart has changed over time and it's become less efficient, right? And we'll talk about that Thursday. Thursday, we're going to talk about changes in the heart that can make the heart less useful. Like if I don't have as much chamber size, you know, how can we maintain stroke volume? Or if my heart is too weak to contract, how can we maintain proper stroke volume? And sometimes it's one factor that has changed. Sometimes it's multiple factors that have changed. That's what does make the heart a little bit complicated, is everything's dynamic. Everything's constantly changing all the time. So you can't ever just look at it at one simple point in time, because while something's leaving the left ventricle, we might be at the exact same time altering our venous return by increasing sympathetic innervation just to the veins, you know? So it does get a little bit complicated. So we're just going to hammer away at the basics, just hammer at the basics. Let's go ahead and take a break. It is about that time. Let me pause the recording here. All right, welcome back. We just took a little quick break, let our minds stretch, wander a little bit, and think probably a little bit of frustration today. Not for me. I could feel it through the computer that there's a little bit of confusion probably going on with the cardiac output and all the factors that get involved with that. So real quick, when we pick back up again, we're going to talk about cardiac output equals metabolic demand and cardiac output equals venous return. Just know that, when we get to the pathologies, that's what will bring the normal physiology to life. That's always what makes it more understandable, because even if you're hearing this stuff for the second or third time, it takes a while just to get the most simple, basic things down. And the reason I call it basic isn't because I think it's easy. It's just there's a certain number of steps we have to go through to talk about normal heart function. And once we have seen all those things, we can then apply that information all across the board. So things like coronary circulation, we kind of have to talk about heart action, stroke volume times heart rate, how the heart works to provide blood to the body. And if you're hearing it for the first time in a different way, it all seems brand new again. And that creates a little bit of frustration. So as we talk more and more about these things, I'm going to repeat them as much as humanly possible. That's all anatomy and physiology really is. It's just repetition. And when you get to your job in the future, that's also going to be repetitive and repetition based. So to understand it better and to acclimate to your environments more, the more basic knowledge that you have, the easier it'll make it for you. But I promise this stuff will click at some point in time in the future. It just takes time. So one of the things that we've been talking about a lot, we did our normal circle diagram of the heart, right? And we talked about blood flow through the heart, just kind of knowing the steps that we need to go through to get circulation going around. That was the first thing that we talked about. So we labeled all these things. It went pretty fast, absolutely, for sure. But just knowing the different chambers, valves, what they're responsible for helps. Being able to recreate that helps. The next thing we talked about was cardiac output equals stroke volume times heart rate. And that's a hang up a lot of times because there's so many different variables that get involved with this. But it all comes down to certain basic phenomenon. Your heart has what's called a basal metabolic rate. And that's the essential, most minimal heart action with no modulation whatsoever. And if you don't have any nerves going to the heart, we can't modulate it, right? So if we were to sever any type of sympathetic transmission, either through the blood or the nerves, and we were to sever the vagus nerves going to the heart, your heart would still beat at about 100 beats per minute. Stroke volume would be pretty minimal. You'd be getting somewhere from 5 to 7 liters per minute going out to the body. But at rest, we don't need quite 7 liters going out to the body. So we want to slow it down a little bit. So some of the factors that get involved with determining cardiac output, again, is parasympathetic versus sympathetic. So we talked about that at a different time. We talked about that before we got into these, quantitative and qualitative descriptions. But when we discuss things like what's input that the medulla needs to be able to properly regulate the heart, and when we're trying to properly regulate the heart, how do we send that there, right? So let's say the medulla is the control center. This works just like any homeostatic function does. Heart's part of the body. Body's trying to maintain homeostasis at all times. Usually we have some type of control center that's going to be monitoring what's going on in the body and can create change that can take us back toward homeostasis. You're going to have some type of receptors receiving information and taking it to the control center. And then you're going to have some type of effectors involved. And that's essentially how everything works in the body, right? And it's controlled through, again, the hypothalamus maybe initially, but when you get into the heart itself, a lot of our input and our outputs coming from the medulla oblongata. And the type of input that the medulla needs to properly regulate the heart is to know, well, how hard is the body working already? How much metabolism is going on? Because any oxygen spent, I need to replenish, right? And any carbon dioxide that I've generated, I need to get rid of, because that's going to alter the pH of the body. So that's a lot of the input that the medulla needs to make these determinations. So I can alter the basal metabolic rate to the appropriate rate of what the body needs. And that is actually described in this formula, which is cardiac output equals metabolic demand. So all the formulas work hand in hand with each other, whether it's the quantitative formula, cardiac output equals stroke volume times heart rate, whether it's the qualitative formula that tells you why the heart needs to be regulated as it is, like why do we alter these numbers? So this equals this all the time, right? And the metabolic demand is oftentimes determined by chemoreceptors, like what chemical changes are going on in the body that let me know what the metabolic demand actually is. So again, if cardiac output has to equal metabolic demand, how do I know what metabolic demand is? CO2 tells me a lot of that, okay? But again, we don't want to send output to the heart that's going to make the heart burst. We don't want to send output to the heart that's going to make, the aorta too pressurized. We also don't want to send out too little output that blood starts to get backed up in the veins. So baroreceptors come in there too. So that's also useful information for the medulla. But regardless, one of the big things, again, that the medulla is looking for all the time in trying to make sure that our cardiac output is equaling metabolic demand is it's just looking for CO2 and hydrogen ion levels. So that usually is your primary input, your primary control, okay? And once we can tell what the body's needs are, again, we call that the metabolic demand of the body, we can then properly regulate cardiac output by altering these two things, right? I can either increase or slow down the heart, which is pretty simple because I can use sympathetic innervation in the form of norepinephrine, and that'll speed the heart rate up, or I can send parasympathetic innervation in the form of acetylcholine through the vagus nerves, and that can slow the heart rate down. But not only can I alter heart rate up and down, I can also alter stroke volume. And stroke volume is the little bit more complicated one to understand. That's basically the amount of blood that's being pushed out per beat, right? So every time the heart beats, how much blood is actually leaving, that's the stroke volume. And one way we can alter stroke volume, like one way I can increase stroke volume is by increasing contractility. That will increase stroke volume. Another way I can increase stroke volume is to distend the ventricle more or increase what's called the preload. And a lot of times we can increase preload by increasing our venous return, okay? Which leads us into another formula of cardiac output equals venous return, okay? So this one ends up being a very applicable type of description of cardiac output, where at first it seems like it's a limiting factor. Basically what this says is I can't push out any more than I receive. But at the same time, if I receive more, I can push out more. So let's say a patient is hypotensive, right? What's one way that we can increase pressure in the vessels? Well, let's just vasoconstrict the vessels. And because veins end up being what's called venous reservoirs, they hold more blood than the arteries do. If I vasoconstrict my veins, that'll send more venous return to the heart, right? Which will increase my preload, right? Which can then increase the stroke volume, getting more blood out into the arteries, right? Which is essentially going to then take my pressure back up again, right? So again, sometimes this seems like it's a limiting factor, but it can actually be a contributing factor to fix it if it's broken, right? But anywho, again, other formulas other than stroke or cardiac output equals stroke volume times heart rate was cardiac output equals metabolic demand. Metabolic demand is just the energy needs of the body and all the stuff that we need to make energies in the blood. So the faster I can get blood to the cells, the more metabolism that they can do, because I need to deliver oxygen, I need to deliver nutrients, And the way I determine how fast I need to get it there is basically how quickly have they burned through the ones they received before. So a lot of times what tells me what my metabolic demand is the byproducts or the metabolites that are generated after we use these things. Because when you take stuff, like let's say this is a cell, like just a normal cell, and you take stuff like O2 and say glucose, and those both enter into a cell, then you can run things like glycolysis, right? You get into the mitochondria, which are O2 dependent reactions, and then you can run things like Krebs cycle and electron transport chain. And as we're using that O2 and that sugar, generate it as a byproduct of that, of course, we get ATP for the cell, which is the metabolic demand of the cell. But at the same time, you're kicking out CO2 and hydrogen ions. Like CO2 is going to be generated by the Krebs cycle. Some of the hydrogen ions are going to be coming from the electron transport chain. And this tells me how quickly cells are burning these nutrients. So that's the type of input that tells me what metabolic demand is, right? And if this goes higher, then my cardiac output needs to go higher because I need to get rid of this faster, circulate it out of the body quicker, right? Or maybe to the kidneys faster, to the lungs faster, and I need to get fresh stuff back to the cells again. So as the metabolic demand goes up, cardiac output also has to go up. How do I know what the metabolic demand is? Usually by the waste that the cells are generating, because the harder cells work, the more ATP they have to make, the more waste gets generated, right? And we have to get rid of that and at the same time replace nutrients and oxygen, okay? So that was the other one. And again, the other one that's very useful to us, we'll just name it again, is cardiac output equals venous return. This was, again, called the Frank-Starling law or the Frank-Starling mechanism. And again, I gave you numerical examples of that. So if I have heart at rest and this left ventricle, let's say, holds 135 milliliters, and under normal contractile properties, we're not going to completely compress all that out. Only a little bit of it's going to come out. We might kick out about 70 milliliters, 65 stays behind. So this was the end diastolic volume. This was the end systolic volume. If you subtract that from that, that gives you your stroke volume, which is kind of duh. But so that's end diastolic volume, that's end systolic volume. If you subtract end systolic volume from end diastolic volume, that's stroke volume. So stroke volume, what is it? It's the amount of blood that gets pushed out per beat. So again, we know that already. And normal, if nothing changes in between, whatever left is usually also what's entering, right? Because it pushes the blood that much further. So let's just pretend this is the body. One misconception, and I know people don't think this in reality, but your mind maybe just doesn't think about it at all. Sometimes when people think about blood flowing through the body, they think every time the heart beats, it's just blood going all the way back, right? Or all the way back. And it's not. Blood's just going a little bit at a time, being pushed forward, push forward, push forward, push forward. So usually if 70 milliliters is leaving, 70 milliliters is entering again, right? But if I push 90 out, that's going to push it a little bit further forward, right? So if I got 70 milliliters leaving, then usually 70 milliliters comes back in. But if I'm pushing out 90 milliliters, usually 90 comes back in. But I can alter this separately. I can have 90 come back in after 70 left, and that can preload the heart more, distend it more, right? You can do that. It's okay. So again, I know there's confusion, but it's okay. We'll get there. And another thing we're going to talk about real quick, we're going to switch gears. I'll open up my iPad right here. We're going to talk about normal blood flow to the heart itself. So one thing we just got done talking about is cardiac output has to equal the metabolic demand of the body. So your heart is responsible for delivering blood to your entire body to meet all of its metabolic demand. But the heart itself also has metabolic demand, and it's relatively high because your heart is one of, if not the most hardworking structure in the body. You know, your heart, your brain are working constantly, right? So they don't get to take breaks. They don't get to stop, you know, every time you're resting like skeletal muscles might, right? So your heart has a pretty constant metabolic demand, even at rest. And when you're doing activity, it becomes greater and greater and greater. So we need good blood flow to the heart itself. So when you're talking about heart circulation, like blood flow to the heart itself, right? This is where coronary circulation comes from. Coronary circulation. So sometimes you might have heard the old saying, somebody's having a coronary. That used to be like an old word for like a heart attack, and it basically meant some of the coronary arteries are broken or clogged or busted. Something's wrong with them. But coronary circulation, in a nutshell, we don't want to get any more complicated than the basics. Let's say this is the heart right here. And so we'll draw it a little bit more anatomically correct. Here is, let's say, your pulmonary trunk. I'm just going to kind of draw like a dotted line. Here's your aortic arch coming up behind it. So the pulmonary trunk's in front of the aorta. Aorta is behind the pulmonary trunk. But your aorta is kind of coming from the left ventricle, this direction. And before we get out into systemic circulation, where you're going to have like your brachiocephalic trunk and your left common carotid and your left subclavian, and then all the blood going down to the lower body through like thoracic and abdominal aorta, you got to supply the heart itself. So two of the first branches that actually come off the aorta right here are your left coronary artery and your right coronary artery. Okay. So this is my left coronary artery. That's an O. And this is your right coronary artery. And there should be a picture of that in the notes. Let's see. Let's get back here. If not, I'll pick one up for us. I thought I put one in there for you. There we go. So here's a picture of that. But when you get into heart circulation, left and right only takes you so far. So my left coronary artery is mostly responsible for the anterior aspect of the heart because it's going to have two major branches. The first major branch of the left coronary artery is going to become the left anterior descending artery, or you can call it the anterior interventricular artery. Right? Same thing. It's the same thing. That's just an AKA for it. So your left coronary artery, its major branch, ends up becoming the left anterior descending artery, also known as the anterior interventricular artery. And it basically descends in what's called the anterior interventricular sulcus. There's like a little groove on the front of the heart, and it kind of goes off at an angle like this. That's how you know you're looking at the front of the heart, because it's going off at an angle, and that's where the vessels kind of ride along. They ride along in that groove, right? This little fake groove I put up here is the groove that kind of exists between the atria and the ventricles, right? So this is sometimes called an AV sulcus or a coronary sulcus, okay? It's just between the atria and the ventricles. But your main division of the left coronary artery is the left anterior descending artery. So your left coronary artery is primarily responsible for the anterior ventricles, which is the majority of the heart, right? The ventricles, so the anterior side of the ventricles, and the left atrium, okay? While your right coronary artery, its main branch is going to actually circle around the back side of the heart, and then it's going to descend in between the two ventricles on the posterior aspect. So the main branch of your right coronary artery is the right posterior descending artery, also known as posterior interventricular. Okay. And then it's also going to have a little branch that'll kind of stay in this groove and supply the right atrium. So right coronary artery, think posterior ventricles and right atrium. And if that's too hard to remember, then let's simplify it even more. Here's our heart, here's our aorta, here is our left coronary artery. What does it do? It mainly supplies the heart on the front side. Here's our right coronary artery. What does it do? It mainly supplies the heart on the backside. So you can't get any easier than that. So it's not so much about left supplies left and right supplies right as left supplies the front of the heart and the right supplies the back of the heart. That's more realistic. Okay. But you do have a circumflex artery that'll supply the left atrium up here. You have a marginal artery that supplies the right atrium up here. But what's the majority of the heart? Majority of the heart's ventricles. Okay. So if I'm going to have a blockage of an artery, what's going to be the more likely places to be blocked? Probably ventricles, right? It's just like if I'm throwing a dart at the heart, what am I most likely to hit? Ventricles, right? So let's think about it. Left coronary artery supplies mostly the front of the heart. Right coronary artery supplies mostly the backside of the heart, okay? And why does the heart need blood supply? It's a very metabolically active organ, right? It's very metabolically active, right? It's always working, always contracting. So you always need that oxygen. You're always producing carbon dioxide. You have to get rid of it. So it never takes a break. And if I can't supply blood fast enough to an organ that's metabolically active, then what happens, right? That's where those words come up. If I can't provide enough blood to a metabolically active organ, you'll get ischemia and possibly infarction, right? So that's why heart disease is such a big deal, because when you get into heart disease, and we'll talk about this more Thursday, heart disease usually refers to coronary artery disease, or CAD. And when you get coronary artery disease, your coronary arteries get fatty plaque buildup inside. That'll decrease the size of the lumen. So ends up looking something like this. And the vessels themselves become more fragile. They're easier to tear. There's more likelihood of developing a clot because the inside has changed. And when blood sees a foreign surface, which this starts to look very foreign to it, it starts to clot. So if you already have a decreased or more narrow lumen, the opening of the vessel, and blood's more likely to clot, then you're going to have very poor circulation past that narrow point, right? So if I have, like, let's say, an increased metabolic demand and I'm trying to increase the amount of blood that gets there, it's going to be very difficult for blood to get there, right? Or if something ends up blocking that, then it doesn't really matter what the metabolic demand, no blood's getting there. Either way, that would lead to some type of ischemia, which is not enough blood to meet metabolic demand. And you'll feel pain in the chest, just like we feel pain in our skeletal muscles when we're using muscles faster than we can replenish the blood. You feel that lactic acid buildup. Well, the heart feels that too. We just call that angina, right? So if the heart is not getting enough blood to meet metabolic demand, it'll usually let you know by hurting, and that's indicating that we have ischemia. And if the blood flow doesn't return back to normal soon, right, or we can't get more blood there soon, then that could lead to an infarction, because an infarction is the same thing. It's just prolonged ischemia. And tissue death occurred. Right? So that's a big deal, because if my heart has atherosclerotic plaque buildup in that main branch, that left anterior descending artery, and the heart's working harder, faster because we're doing exercise, I'm not able to get blood there fast enough to meet the metabolic demand of my heart itself. So you'll have chest pain, right? And again, not only am I narrowing the lumen of that vessel, the vessel itself is changing inside, which is going to make it more likely for clots to form. So if I already have a narrow opening, you're going to know it every time you're doing activity, because the activity is going to stress the heart out, which is going to lead to more metabolic demand, which means I need to have more cardiac output. But because I'm not receiving enough blood, you're going to become ischemic. You're going to start building up lactic acid. You're going to feel chest pain or angina, right? But if we don't get the blood there soon, then the heart can actually die. Anything distal to that site of blockage, right? And when we have changes, again, a clot can form. It can totally block this off, and basically everything distal to that will die, right? So that's what we call a myocardial infarction, right, or heart attack. So we'll talk about that more on Thursday when we get into the pathologies. But this is how it starts for a lot of people. Still, one of the most common ways that we are dying in this country is heart disease, for sure, which is basically coronary artery disease, which sets us up for more likelihood of having heart attacks. And when you damage heart, the heart's going to change over time. So that usually leads to cardiomegaly. And cardiomegaly always leads to congestive heart failure, right? But you can get this without this. You don't have to have that. You don't have to have this to get cardiomegaly and congestive heart failure. You can just have hypertension all the time. So we'll discuss all these. Again, we'll discuss all those. And it helps you understand the basics better. When you understand the pathology, then you understand the normal physiology better. It helps drastically. So that's blood flow to the heart. Let me see, what else was I going to talk about today? Oh, conduction system. Let's do quick conduction system. So let's just draw circle diagram again. Conduction system, your heart let's see. Think about it as the depolarization flow across the heart. And the heart itself is self-excitatory. And throughout the heart, we have these specialized cardiomyocytes that can depolarize on their own without any input whatsoever. Like you don't need a neuron to tell it to fire. You don't need a certain type of neurotransmitter to tell it when to fire. It just fires on its own based off fluid electrolyte normal values, right? And wherever we fire first is going to be our kind of lead node. And from that point, it's going to spread out from that. And in most people, that the easiest part to depolarize or the fastest portion to depolarize is up here in the atria called an SA node. Right. So that is usually what is most people's pacemaker of the heart, right? The SA node, we sometimes call the pacemaker. And some people are born without an SA node, right? And they usually would have a slower heart to depolarize because the heart, again, is self-excitatory. The atria can contract on their own without an SA node, but usually not as fast. This is the fastest place to, depolarize in the heart, so that's usually where it starts, and it can go from there. But you can be missing an SA node, you can have a broken AV node, and your ventricles contract on their own, like the heart itself down in the ventricles can contract on their own, but it's, again, much, much slower. So normally, in most people, we're going to start with an SA node. That signal is going to fire. It's going to ride across the atria. Eventually, that signal is going to reach something called an AV node. And that AV node is kind of located within the wall of the septum. Like this is called an interatrial septum. This is called an interventricular septum. They sometimes call this the inner skeleton of the heart, made up of like cartilaginous fibers and thick muscle and connective tissues. And when you enter from the SA node to that AV node, the signal slows down a little bit. So as the atria are receiving signals from the SA node, the atria are going to fire first, and we'll talk about why that's important in a minute. But that signal is going to consolidate down to an AV node, and from the AV node, we're going to thicken up a little bit because that's going to help speed the signal back up again. And you end up getting to this thing called an AV bundle, or the AV bundle of Phys. And it's going to speed that signal up. So as the signal comes in here, it's going to slow down a little bit. The AV bundle of Phys is going to speed it back up again. But still, we're going to already have a nice little lull between the atria firing and eventually the ventricles firing. From that AV bundle of Phys, you're going to have bundle branches that travel down the interventricular septum, so kind of within the walls of the heart itself. It's going to go all the way down to the bottom here, and it's going to fire up Purkinje fibers in this direction. So as we're going side to side down here, these are called bundle branches. And these bundle branches will lead it to the Purkinje fibers that are here at the bottom. So that's your basic cardiac conduction system, right? You have an SA node, an AV node, an AV bundle of His, or sometimes they just call it the AV bundle, or sometimes they just call it the bundle of His. You have bundle branches, and you have Purkinje fibers. And again, these are self-excitatory, like the SA node will fire on its own without any control coming from neurons, right? And it's based off fluid electrolyte volume. Specifically, the number of dissolved sodium, calcium, and potassium that's floating around in the blood and within the cell. So this is why it's very important that a person has proper, blood, sodium, calcium, and potassium. Because if the blood levels of these electrolytes are off, it's going to mess up this normal excitability of the heart, right? Which is based off sodium slowly dissolving in, or not dissolving, but slowly diffusing into the heart cells, calcium diffusing into the heart cells, potassium leaving the heart cells. And so if it's having to go against too strong a gradient, or the gradient's not strong enough, that's going to change the rate at which these things will fire. So the heart can go either too slow and fire very weakly, or the heart might go too fast, or the heart might contract and have a difficult time relaxing. So when a person's hyper or hyponatremic, calcemic, or kalemic, heart is a big deal, right? It's going to affect the heart. But let's say I want to make the SA node more responsive to these things, then maybe sympathetic innervation can speed this up. Or maybe if I want to make the heart less responsive to some of these things, then maybe parasympathetic innervation can slow them down. So we can then further modulate the heart. So they are self-excitatory, but I can modulate how excitable they are with sympathetics versus parasympathetics. So if I want to speed that rate of depolarization up, let's add some norepinephrine. If I want to slow that rate of depolarization down, let's add some acetylcholine. Okay, so normally at rest, your vagus nerve is sending your SA node acetylcholine just to make it less responsive to these things, just slightly, so it slows the heart rate down. But if you have a vagal response and the vagus nerve sends too much acetylcholine, it'll slow down too much, right? And the heart might stop beating for a little bit, and then you can pass out. So hopefully we'll talk about that later. So that's a conduction system. And one reason we're talking about that conduction system now, and one reason we want to separate our atrial synctium versus our ventricular synctium, like when the atria fire, they fire together. When the ventricles fire, they fire together at the same time. We want to separate those two because there's something called atrial systole that we're going to have prior to ventricular systole that's going to help prime the heart so it can beat better. So we're going to talk about that now. And one thing that we mentioned to you earlier before was the term systole versus diastole. So here's where I'm really going to throw a wrench in the gears. Okay. When you think of the word diastole, you're thinking about the refilling phase of the heart. And the main thing you're thinking about is refilling of the ventricles. So how does that work? Let's draw our circle diagram. So during diastole, certain valves should be open, certain valves should be shut. So if I'm trying to refill the heart, and specifically the ventricles, right, then my AV valves should be open, right? My semilunar valves should be shut. And real quick, just in case you need a refresher on this, a lot of times when I'm drawing valves, I draw valves like they're hinges, like they're doors on hinges. So if you, let's say we're in a leg vein. Let's say this is a leg vein. And again, I'm just talking about valve function or valves. Valves serve two purposes. right? Two purposes. A valve, a healthy valve, allows a single direction of flow, and once it closes, it prevents backflow. Okay, it's important to remember, because most of the time when you talk about valves, you think about this part, it prevents backflow, but if it doesn't open right, that's no good either. So you need a valve to be able to open and close adequately. So valves kind of work based off pressure differentials on either side of the valve, and think about it like a hinged door, right? So if this is the little door right here, and this is another little door right here, if I have more pressure on this side than I do this side, then that'll push the valve open, right? So the valve will then open this way and blood is allowed to pass through, okay? But now let's say blood tries to drop back down. That's why I'm drawing in a leg vein, because we're going up and it's just easy to visualize like this. But let's say blood starts to push back down again. This valve should then close, right? And that will prevent the backflow, right? So a valve has two functions. We need to allow a single direction of flow, so blood goes in the proper direction, and it should also prevent backflow. So when I'm drawing that picture in the heart, if the little leaflets are open like this, that means blood's going through, the valve is open. If the leaflets are closed like this, that just means that's a shut valve. So during diastole, the refilling phase of the heart, we're mainly trying to refill the ventricles. And as blood's coming in, this is very passive at first. So if I'm getting my venous return of the body, or if I'm getting blood coming back from the lungs through pulmonary veins, this is all happening at once. You know, when we're tracing a drop of blood, we like to go through one chamber at a time, one side at a time. But, you know, when blood's flowing through the heart, you know, it's all happening simultaneously. So as venous return's coming in here and, you know, pulmonary veins are bringing back blood over here, blood enters into the atria, and that'll immediately increase pressure inside your atria. And because your ventricles had just pumped a minute ago, there's not as much blood in here. There's very little blood in the ventricles, so the pressure is low inside of here. So as that blood enters and the pressure in the atria starts to rise, that'll open these valves up. So basically, just think about it. The weight of the blood is enough to open the valves, right? So these valves will open and blood will just passively enter into the ventricles. So how long does that occur? How full do we need to get before that no longer happens? So during diastole, when we're refilling the ventricles, the first 70% of diastole is passive, meaning as the blood's entering into the atria, it'll just passively go into the ventricles without any help or any assistance whatsoever, because as blood enters the atria, that's going to increase internal atrial pressure. It's basically the weight of the blood itself is enough to open these valves, and the blood just passively enters. But once we get about 70% full in the ventricles, pressure in here will start to equalize the pressure that's in here. So your valve will have no benefit going one direction or the other. So it just simply prematurely closes, right? So at 70% full, AV valves will close, okay? But are the ventricles primed yet? Is there any distension going on? Is this going to be a very adequate contraction? You know, if I'm only 70% full? No, that's why we said it's only 70% full. So it's only 2/3 as full as it needs to be, right? And to have a good, efficient heart, we need this to be at least 100% full, right? Even though we can go more, right? So I want this thing to be 100% full, but this valve is already closed. So how are we going to get the remaining blood that's still entering into the atria, into the ventricles? We have to go through what's called atrial systole. So we will go through atrial systole. And that will happen because the SA node is going to fire first. And when that SA node fires, your atria are going to, contract. And when the atria contract, that's going to compress that blood that's left over in these atria. It's going to open these valves back up again. And that last 30% of the blood is now going to enter. Okay, so because of atrial systole, we have now filled our ventricles. So right after that happens, the signal is going to be traveling through the heart, right? and the ventricles are then going to contract. And the first thing that's going to happen when these ventricles contract is we're going to slam shut the AV valves, which is going to give us our first heart sound. So as the ventricles contract, you know, they're 100% full now. So the pressure in here is already high. The ventricles contract, which really spikes the pressure. These AV valves will slam shut again, right? And that's going to generate our first heart sounds. But now that the heart has built up all this pressure, we're able to overcome what's called peripheral resistance, which is usually in the aorta about 90 millimeters of mercury pressure. Once we've generated enough pressure by filling this ventricle up with fluid, it's 100% full now, and now we've gone through what's called isovolumic contraction. Don't worry about that word. It just means I'm going to contract on the fluid that's inside of here. That slams that valve shut. The pressure in here spikes to about 120 millimeters of mercury pressure. Now it's now able to overcome peripheral resistance, which is always pushing back. So now your semilunar valve opens, like your aortic valve, and blood is able to exit. And once the blood level in here drops down and we start to go below 120 millimeters of mercury down to 110, 100, 90, 80, eventually as the fluid leaves, peripheral resistance stays pretty the same, right? This isn't going to really change. Once this number becomes greater than this number, then this valve will shut, right? This valve will then open because blood is left and the pressure in here is decreased, and we'll start refilling again, right? So let's do it one more time. So during diastole, this is why I always start with diastole first, because you got to have something to work with. During diastole, the main objective of that is to refill the ventricles. And most of the refilling of the ventricles is pretty passive at first, right? Because you got venous return from the body coming in, you got pulmonary veins dumping blood back into the other side. And as the blood enters into the atria, the pressure inside the atria becomes higher than the ventricles because the ventricles have already ejected blood, right? So you have passive refilling that occurs until the heart's about 70% full on each side. Once you're at 70% full, right, pressures will equalize between your ventricles and the atria, and your AV valves will prematurely close, okay? So what that means is we still have some blood left over in the atria. We still haven't finished filling the ventricles. So in order to finish filling the ventricles, or you could say finish emptying the atria, then we need to go through atrial systole. So what is atrial systole going to do? Finish emptying the atria, finish filling the ventricles. So that's going to compress this blood, forcing that valve back open again. The last 30% is going to now come in, which is going to fill this thing up to 100%. 100%. Now the pressure in here is already pretty high because it's full, right? And that fluid's pushing on the walls equally. Then we're going to have what's called isovolumic contraction, where the heart's just going to compress that fluid that's in there, not letting it go yet, but just compressing the fluid that's in there. That's going to slam your AV valves shut. And eventually we're going to overcome peripheral resistance heading out this direction. So your semilunar valves are going to open, and then you can eject the blood, and then that becomes your systole. So during systole, AV valves have slammed shut, preventing backflow. Semilunar valves are open, right, allowing the blood ejection to take place. But once the blood has left, and there's very little blood left in here, the pressure in here will drop. Which will then allow back pressure to shut the semilunar valves. So the back pressure then overcomes semilunar valves. That shuts those. And because we don't have as much blood left in here anymore, the pressure is lower in here, and blood is filling back into the atria, which is increasing pressure in there. So that allows us to go through diastole. Easy peasy, lemon squeezy. That's enough for today. That's plenty of stuff to think about. And again, there's quite a few pictures in the notes that goes over some of these things. We've actually covered quite a bit of this. I'll talk about this more on Thursday. This is actually putting everything together. But another big thing that we want to do Thursday is pull out a lot of this pathophysiology, like what would changing the shape of the heart do to the heart, right? What happens if a valve isn't closing adequately, or if we can't get into the aorta adequately, right? Or what if peripheral resistance is too great for the heart to overcome? What if the heart has been weakened by some type of ischemia, right? So all that kind of stuff, it's all in the notes, everything that we've kind of talked about today, but it's mixed in with the pathophys. So today we kind of did all the normal physiology, right? So here's conduction basics, all that kind of stuff. This was a big discussion point today. Another big discussion point was things like this and this, factors that affect these things. But Thursday, we're going to talk about how it breaks. You know, like, again, if I don't have enough blood going through coronary arteries, heart attack. You know, if I have a heart that's too big and too stiff, how is that going to affect stroke volume, right? If I have a heart that's too big and too floppy, how does that affect stroke volume? And if we don't get proper stroke volume and blood starts to back up, what does that cause? So that'll make all this start to come more to life. But again, it's a decent amount of information. So read through it, bring any questions that you might have, because there's always questions about the basics of cardio phys. Bring that Thursday. But after we get done Thursday, things will start to make more sense. Okay. So let me again show you where we are. Today was our Zoom meeting for 620. We just got done going over a lot of the basics of cardio phys. Thursday, we're going to talk about when it breaks, what happens to it. And then, you know, if we don't get finished with it, we'll carry it on into the next week. But next week, the way it's looking, we'll talk about blood flow and pressure. That should be pretty straightforward and simple. Respiratory system and gas exchange should be pretty straightforward and simple. Once you have some of the tools that we gather in cardio phys, these become a lot easier. So we can even use time next week to talk more about this if you have questions. The week after that, one of the days we don't have a class meeting because of July 4th. The other day is a Zoom Q&A. So again, we can answer any questions that you want about cardio. So don't hesitate to ask about cardio, because this is going to be one of the most useful chapters that you ever use. So if I said anything that makes no sense at all, stick around, make sure you ask me questions about it. Or if you are listening to this recording later and you have any questions, then bring it with you for our next meeting too. But if you got questions anytime in between, email me. But that's enough for today. So on Thursday, we'll talk about some cardiopathophys. So have a good rest of the day. See y'all on Thursday. Thank you. Thank you.